Log on, tune in, find out. Another good idea from Cambridge.
Well, a very warm welcome to this sixth in the present Darwin College lecture series on the subject of life. This week we're turning to the tiny building blocks of life, cells. How do they know what role to adopt in life? How do they know how numerous to be? And how do they know when to die? The subject is the life and death of a cell. Ron Lasky, the speaker, recently retired as the Charles Darwin Professor of Animal Embryology and also as the director of the MRC's Cancer Cell Unit at Addenbrooke's. And his research has been concerned, among other things, with cell proliferation and why, in particular in cancers, it goes wrong. Ron's won many international accolades for his work, but I think the most distinctive is for his albums of songs. Songs for Cynical Scientists, strongly recommended. And here, alas, without guitar, is Ron Lasky.
Thank you very much. My topic tonight is life and death of a cell. So first I should apologize to all those of you who've come to hear a lecture on prison reform, death in police custody, or design of medieval monasteries. You won't. Instead, I'm going to talk about the living cells that make up our bodies. And most of you will be aware that in the last year, the number of human bodies on the planet is estimated at reaching the staggeringly large number of 7 billion. None of us has difficulty in recognizing that 7 billion is a truly enormous number. So it may therefore surprise you to realize that 7 billion is the number of cells in the tip of my index finger. So that's probably a slightly inaccurate estimate. It's probably nearer 9 billion, but it's of that order. So the human body consists of 100 trillion cells, or 10 to the 14 in new money, and all of those have to be regulated in ways that generate a functional body. So an inescapable conclusion is that cells are very small, with certain exceptions, such as this ostrich egg, which starts life as a single cell. But leaving that aside, I'm going to start by trying to infect you with enthusiasm for the beauty and fascination of the cells that make up our bodies. And I'm then going to move on to look at cell division and how all cells arise by division of pre-existing cells. I shall then try to convince you that death can be good for you when it's cell death, and that cell death plays an important part in regulating the form and function of our bodies and in sculpting the design of our bodies as well. I'm then going to look at the challenge that cell division and cell death, working in opposite directions, poses for maintenance of the body. So if our bodies have constant cell division going on and constant cell death, how on earth do we maintain the appropriate balanced number of the various different types of cell in the body? This challenge is made much greater by the fact that the rate of turnover of different types of cell is fundamentally different. So the cells that line our intestines are being renewed every few days, whereas most of our neurons are with us throughout our adult life. So how on earth are these different turnover rates coordinated and balanced? I'll then look at what goes wrong in this balance. For example, excessive cell division with inadequate cell death results in cancer. Conversely, excessive cell death without compensating cell renewal contributes to the range of neurodegenerative diseases which are becoming increasingly tragic as the population ages. So these are the topics I'm going to consider. I should reassure those of you who were here last week that I shall not be showing anatomical details with the exception of my left hand, and with the exception of microscope images of cells and groups of cells. So I shall be showing clinical specimens, but they will be microscope images rather than full-scale images. So to start by looking at the living cell, well, here's one my son prepared earlier. So this is not actually a living cell, it's a pickled cell, which has been pickled to preserve its structure and then stained so that components of the cell are stained 3 different colors. DNA in the nucleus in the center of the cell is stained blue. This is the information archive of the cell, which I'll have much more to say about in the next few minutes. The power stations of the cell, so-called mitochondria, are stained red. These are responsible for the energy transactions. They convert the foodstuffs we eat into a form of energy that is available throughout the cell, but they also play a role in regulating cell death. The third component shown here are stained in green, are components of the cell's skeleton, microtubules. These are highly dynamic rails along which cargoes can move in either direction. For example, the clustering of the mitochondria, the power stations here out at the periphery or around the nucleus, is largely because they can be transported along microtubules, as can the various other components of the cell, which are not shown in this image, but of which there are very, very many more. So the microtubules, these green skeletal elements, will feature prominently when we come to look at cell division, as they are an essential part of the mechanism of dividing the cell in 2. So first then I'm going to look at the nucleus and DNA. And I should apologize to the biologists in the audience, of which I know there are several, on the grounds that I'm very deliberately going to start this lecture at a level that is not accessible only to biologists. I'm very deliberately going to aim to ensure that as many of you as possible can have a chance of understanding what I'm saying. So I'm going to start then with the nucleus and the DNA that it contains. With the aid of this visual aid of the cycle path that runs from Addenbrooke's Hospital, seen there, parallel to the railway, from Addenbrooke's to Shelford. And it illustrates the structure of one gene. It's the gene which encodes a breast cancer susceptibility gene called BRCA2, or BRCA2. And the 4 letters that make up DNA, A, C, G, and T, are illustrated in these 4 colors. A is green, T is red, C is blue, and G is yellow on the footpath. And these encode the structure of the proteins that the cell produces. They constitute, these base pairs constitute the rungs, the horizontal rungs of the double helical ladder shown by this sculpture at the start of the path. So these are the building blocks. What they do is they're copied in the nucleus into RNA, which carries the word out into the cytoplasm, where it's translated into proteins, and the proteins then build the structures and do the work. But the structural information encoded in DNA is transcribed into RNA. RNA is exported from the nucleus out to the perimeter, where it's translated into protein, and the protein does the rest. A further point is that DNA is copied to make more DNA each time the cell divides. It has to first double its DNA, and that, of course, all takes place within the nucleus. So, how much information is encoded? Well, a key feature of DNA is that it's self-complementary. So as the two strands, to make two copies, the two strands unwind, and each serves as a template for making a copy which was identical to the original strand. And as there are biologists sitting near the front of this audience, I will challenge you to tell me what is wrong with this tie. There is a serious flaw in the design of the tie. But for the rest of you, don't worry about that too much. But the issue is that as DNA is duplicated, so it has to unwind, and each of the two strands then serves as the template to make a second strand in this way. I'm afraid the color code on the tie is different from the color code on the cycle path. There's little I can do to correct that. But how much information is encoded in DNA in each cell? The answer is frankly shocking. This is an illustration of it. This is in the Wellcome Trust Museum, that's the Henry Wellcome Museum in the Euston Road, and it illustrates the human genome present in each of our cells. And it illustrates it in the form of book pages like this, where all of the letters on this page are A, C, G, and T, and that book can be seen here. It's a large encyclopedia-like book, but the startling thing about this slide is that it comes from that gap there in this series of 118 similar volumes, all the size of large encyclopedias, that together make up the information in the human genome. So this bookcase that reaches from the museum floor to the museum ceiling contains 118 such volumes to represent the whole of the human genome, and that is encoded in each of the cells, of the 7 billion of which is contained in my fingertip. It's an extraordinary information packaging and access challenge. I'm going to illustrate the amount of DNA packaged by one further slightly challenging analogy. And it resorts again to my hand. This time not just a fingertip, but if we consider all the digits on a hand, then they would contain about 100 billion cells. Each of those cells contains 2 meters of DNA. That's 200 million kilometers of DNA, enough to reach the sun. So if we extracted DNA from the fingers of one hand and joined it all together, it would reach the sun. So there is a remarkable packaging challenge going on in our hand, or in all the cells of our body, which I'll have more to say about in a moment. But now I'm going to move on to look at cell division. And if there is such massive length of DNA in each cell, the first thing that is necessary for a cell to divide is for that DNA to be condensed down into chromosomes, and that's what chromosomes are. They're condensed DNA coiled up 10,000-fold so that a chromosome is 10,000 times shorter than the DNA that it contains. So those chromosomes can then be split longitudinally and pulled apart on this spindle of microtubules, stained green in this image just as they were in a previous one that I showed you. Just as the DNA was stained blue, it's stained blue again, and the microtubules are stained green again. How this works, I'll illustrate in a moment. But the key point about cell division is that it's essential to produce new cells. The Prussian pathologist Rudolf Virchow, Virchow, Torsten Kruder has been trying to educate me in how to pronounce it, and I apologize to Torsten for getting it completely wrong the first time. But Rudolf Virchow pointed out that all cells arise from pre-existing cells. They don't arise de novo. And The way they do this is by division. This is a later stage of division from the same series that I just showed in the previous slide. And this slide shows it in more detail. A resting cell that is not about to divide has a nucleus in which the DNA is dispersed almost uniformly through that nucleus. For division, the DNA coils into chromosomes, which you can see beginning to happen here and is much clearer by here, and it becomes organized on the spindle of tubulin, still green, even though the DNA is now red rather than blue, and the spindle of tubulin organizes the chromosomes so that they form a flat plate across the middle. Only when they're in a flat plate can they then split longitudinally to be divided. The reason for that is they won't split until they're receiving equal pulling forces from the two ends. Only then are they ready to divide. And the reason for that is it ensures that each chromosome is divided and you don't get two copies of one going into the same cell. So it's only when they're aligned that the signal is given for them to split so they can be divided. They then decondense, and round the cycle it goes again. This looks rather more dramatic in a video, which I'm about to show here. This video goes through fairly quickly. It's speeded up. So I'm going to take you through it 3 times, possibly 4, to see what's happening. So first, the chromosomes will condense. The chromosomes are condensing. They're being organized on the equatorial plate of the spindle. They've separated and been pulled apart. In this next cycle of the same image, I want to draw your attention to the fact that the spindle elongates as they're being pulled apart. These poles have moved away from each other and have elongated and been moved further away. So how's that happening? Well, the easiest way to show you how that's happening is with the aid of a very sophisticated teaching aid, my 60th birthday cake, sadly long ago. And this illustrates the principles more clearly than I can with any dramatic image such as the one I've just shown you. So this was also made by our son, and the chromosomes on this are blue. The microtubules are no longer green. I'm afraid they're red. I hope that doesn't confuse you excessively. But the thing it shows very clearly, apart from the fact that the poles of the spindles are illustrated here in green, but the thing it shows is that there are 2 types of microtubule. There are some that reach from the pole to the chromosome, like this one, or this one here, which reaches from the pole to the chromosome. There are others which reach from one pole to the other pole. These both have functions to play in moving the chromosomes apart. So the first ones are the ones that reach from the pole to the chromosome. And progression of the chromosome up these is very much like somebody nibbling a spaghetti noodle. So the chromosomes literally nibble their way along the microtubule, spitting out the fragments as they go. And so they shorten the microtubule and move towards the pole by depolymerizing it, literally chewing it up as they move along it. That moves the chromosomes towards the pole. It doesn't move the poles apart. The poles are then moved apart by different microtubules, the pole-to-pole microtubules, pushing against each other with the aid of molecular motors that run along them, pushing the neighboring microtubule away, and thus pushing the poles apart by elongating the spindle through molecular motors, pushing the fibrils past each other. So in that way, the chromosomes can be moved right apart into the 2 progeny cells. So the process of nibbling away the noodle and moving up towards the pole is done by a specialized part of the chromosome, seen here, which attaches to the microtubules, and it's only when this part is attached to both poles by a microtubule that the chromosome can split longitudinally down this cleft here, and one half goes to one cell and the other half goes to the other. Now, this is a single DNA molecule in each of these halves. A highly coiled single DNA molecule, as I've told you, it's shortened by 10,000-fold, but it's a single DNA molecule in each of these halves. So there is a logistical nightmare, because before the chromosome can divide like this, each progeny cell is going to get one of these two double helical molecules, which it then has to make a second one and separate those before it can divide again. It's got to duplicate all of this DNA. Now, that poses another horrendous challenge. And the challenge is shown here in numbers that biologists will nod sagely and say, yes, they're comfortable with these numbers. And I'll hopefully make the rest of you more comfortable with them in a moment. But the diameter of DNA is 2 nanometers. The length of DNA in each nucleus, put end to end, would be 2 meters. But the diameter of a nucleus is only 5 microns. Now, what does that mean? Well, to understand what it means, it helps to have a scale model expanded 1 million-fold. And if I give you a scale model expanded 1 million-fold, DNA then becomes the diameter of thin string, 2 millimeters in diameter. And it's contained in a nucleus of 5 meters diameter, roughly the volume above this stage, much less than the volume of this lecture theatre. But the DNA it contains would reach from Newnham to Naples, or from Sidgwick Avenue to St. Petersburg. It's 2,000 kilometers, packaged in a volume the size of the space above the stage. And yet all of it has to be packaged in such a way that it's accessible for regulated duplication, so that it can be duplicated before the cell can divide. It gets worse, because it's not enough to just duplicate it approximately. It's essential that all of the DNA is duplicated completely. Otherwise, there will be a chromosome break next time the cell tries to divide, and that in turn can contribute to an increased risk of cancer. Furthermore, it's not enough to do the same piece twice. If you do that, that also increases the risk of cancer in ways that I don't have time to explain today. But either too much replication, or too much duplication of DNA, or not enough duplication, are both potentially carcinogenic events. It's essential that the cell copies its DNA exactly once and only once. So how does it do this? Well, this question has been a personal obsession of mine for over 30 years, and we understand how it works. This is a gross oversimplification, but this is a model we proposed in 1981 to explain how the cell knows which bits of DNA it's done and which it hasn't. The challenge is that to synthesize all the DNA in the short time the cell has to do this, it starts at 100,000 different locations along the DNA. And yet it has to coordinate those to do all of them once and only once. It's a massive bookkeeping challenge. And it's achieved by distributing a license to duplicate DNA along the DNA. These are the 2 strands of DNA you see running along here. And it distributes a protein license which allows the DNA to be duplicated before any synthesis starts. Once DNA synthesis has started, it displaces the license from the DNA that's been synthesized, but retains it on the DNA that has not yet been synthesized, so that these pieces can still synthesize DNA. These pieces know that they've synthesized DNA. They no longer have the license and can't do it twice. The clock is reset by the act of cell division, which modifies these licenses and allows them to bind again, to go around the board again. It's very much like molecular monopoly. You collect 200 pounds of factor when you pass through cell division, allowing you to go around the board once more. I've oversimplified this, but to convince you that we do know how it works and what the proteins are that do this, I'm going to show you that we can fool it. We can make it go wrong. We can jam this system so that it gets it completely wrong. And this is an example of it. The DNA is stained red, And these are normal cells with DNA stained red. These are the same scale, but after we have confused that system so that it keeps making DNA multiple times without dividing. This nucleus is about 200 times the DNA content of these nuclei because it's kept on making DNA repeatedly without dividing, so resulting in giant cells. So there's one final point about DNA that I want to introduce you to. It's the most challenging part of the lecture, so if you don't understand this, don't give up and rush for the exits. It will get easier after this. But I want to introduce you to some rather mystical activities that exist in the cells. This isn't just to torment you, it's because I want to come back to them because they're relevant to what we can do about cancer. And I want to introduce you to proteins that unwind one DNA strand from the other. Because each cell contains 6 billion base pairs, 3 billion from each parent. It contains 6 billion base pairs, but for every 10 base pairs, the rungs on the ladder, for every 10 of them, there is one helical twist of one strand around the other. Imagine a 2-strand cable, and you start to pull the strands apart, which has to be done to duplicate them. You're left with an awful lot of torsion in the rest of the cable. It passes the twists on upstream. So I want to introduce you later to the proteins that unwind one strand from another, because they turn out to be very powerful diagnostic tools for detecting cancer at early stages. But I also want to introduce you to activities that remove the torsion that's generated by unwinding one strand from the other. They can untwist and relax torsional strain in the rest of the molecule. How do they do it? Well, the way they do it is extraordinary. There are 2 classes I'm going to tell you about. One of them, the class 1 enzymes, make a nick in one of the 2 strands of DNA. They then form a chemical bond to the end that they've generated, so there's a protein-DNA chemical linkage. They hold on tight to that, and they pass the other strand through the gap while they're holding on. So they make a nick and pass the opposite strand of the helix through the nick, and that gradually, one step at a time, it removes the torsional strain from the molecule. The other ones, the type 2 enzymes, are even more mystical. What they do is they cut through both strands of the DNA, and they pass a second double helix through the gap. Now that's wonderful for untangling the final products of DNA synthesis. You can untangle them completely, if they're tangled, by cutting through one and passing the other one through it, and removing strain that way. Both of these mystical activities I'm telling you about not just to alarm you, but because they turn out to be important in terms of either cancer diagnosis or cancer treatment. So they have horrible names. These are called DNA helicases, the ones that unwind one strand from the other. These are called DNA topoisomerases. Don't worry if you haven't memorized them. There is not a written exam at the end of the lecture. And from here on, it will get easier. So I'm now going to try to convince you that death can be good for you. Improbable though it sounds. When it's cell death, it can be important and beneficial. So the bad news is that during this lecture, a few billion of your cells will die. The good news is that a few billion more will be formed. Somehow a balance has to be maintained, and that's a balance that I shall return to in a moment. But Steve Jobs, the late Steve Jobs, who was the intellectual leader of Apple, wrote very perceptively that death is very likely the single best invention of life. It is life's change agent. It clears out the old to make way for the new. Cell death is vitally important. Programmed cell death was co-discovered by the former professor of pathology here in Cambridge, but he discovered it while he was doing his PhD in Aberdeen, Andrew Wiley. I hardly need tell you which of the two he is. But Andrew discovered this in 1973, and for many years was a voice in the wilderness as people failed to take this phenomenon seriously, even though those of us who talked to him about it in the 1970s were completely convinced that it was important. It is important. It's a very dramatic event. This shows a culture of cells. This is a normal, healthy nucleus. This is one that has had a signal to kill itself, and it does so dramatically. Programmed cell death is a phenomenon that is rather like the equivalent of stabbing, shooting, drowning, hanging, and poisoning the victim. It leaves nothing to chance. It makes sure that it is not just dead, it is very, very dead. The rationale for this is that if you have a wounded or damaged cell still able to proliferate, it will accelerate progress towards cancer. You don't want damaged, wounded cells to be able to proliferate. You want only correct, well-behaved cells to proliferate as a defense against the incidence of cancer. So I'm going to show you a video of cell death in stages. It was made partly by Gerard Evan, the current professor of biochemistry, and these two cells shown by the arrow will undergo cell death. It's speeded up substantially, and I'm going to show you this twice. I'm then going to show it in a different way. But you'll notice these cells disintegrate. They literally boil, bubble, and absolutely disintegrate. It's a dramatic process. And I'm now going to pause this to take you through the stages in glorious Technicolor, again with aid of this video from Gerard Evan and Joshua Goldstein. And what it shows is these cells have been genetically manipulated so that their mitochondria, the proteins I told you early on are the power stations of the cell, the organelles that are the power stations of the cell, but are also involved in cell death. They contain a green protein called cytochrome C. It's involved in respiration, but that protein is also involved in triggering cell death, or in amplifying a signal for cell death. And you see there are discrete rods here. But an early event is release of that protein into the rest of the cell, out of the mitochondrion into the rest of the cell, this diffuse green color. Then mayhem sets in. These cells are being cultured in a dye which would stain DNA if it could get into the cell. The fact the nucleus here is not blue tells you it can't get in. But the membrane then gets disrupted as the cell is undergoing dramatic suicide. It's disrupted, and the dye gets in and stains the DNA. Furthermore, another dye gets in which can only stain the inside of cell membranes, red. So the red dye shows the cell membrane is completely disintegrating. It's bubbling, blebbing, and pretty well giving up the ghost dramatically. I'm going to show you a video of those consecutive stages. So here you see the cells with discrete mitochondria, which have now leaked. Now DNA is starting to be stained. The inside of the cell membranes is starting to be stained red, and total mayhem and disintegration and destruction has fallen on the population. I'm going to show it again. Normal, discrete, rod-like mitochondria start to leak, release the cytochrome C, which activates a whole cascade of damaging events, and the proteins of the cell and the DNA of the cell are cut to pieces. It's a very devastating way of ensuring that the cell is destroyed completely and cleanly. So that event illustrates the power of cell death. I'm now going to go back into the program and illustrate one further thing that cell death does for us. Because cell death has another role. How are digits formed in the hand? This is the last time I'll inflict this picture of my hand upon you. But how are digits formed? Well, in the embryo, they start as a broad paddle. So how does it get from being a broad paddle into discrete digits? The answer is programmed cell death. Regulated cell death selectively kills the cells at this point in the paddle, sculpting the digits exactly as a sculptor would carve a hand from stone. They're not generated by growth of the fingers, they're carved out of a flat paddle by programmed cell death at these sites. So death can be extremely good for you. It's an essential part of the maintenance of our bodies. So I'm now going on to look at the challenge of cell renewal, how we manage to produce the right number of cells at the right time. And for this particular purpose, I'm going to start by looking at the rate of turnover of different types of cell. So the lining of the gut, or the duct, the pancreatic duct that carries digestive juices into our intestines, the cells in this very hostile environment last only a few days before they're replaced. Skin cells in a slightly less hostile environment last for weeks before they're replaced. Red blood cells last months. White blood cells either last just a few days, and when the infection is over, for example, they can be destroyed quickly, or they can persist for months to retain the immunological memory to protect you against a subsequent challenge of the same threat. Nerve cells mostly last our lifetime, exception being odor receptors for our sense of smell. Those, of course, are also exposed to the environment, and they last only a matter of a few months. Retinal receptors in the eye or the hair cells of the cochlea in the ear should last our lifetime. Obviously, the problems arise with our limited ability to replace these cells, which should last a full lifetime. So, patterns of cell proliferation are very, very tightly regulated. That's illustrated here because the yellow nuclei are actually a combination of red and green, but what you can see is this precise pattern. The yellow cells are not randomly distributed through this tissue, they're quite precise patterns of distribution. These are actually sections through intestine taken at right angles to each other, and these test tube-like crypts are sites of DNA synthesis and cell proliferation, seen here in section. But the exception to this tight regulation of patterns of cell proliferation is, of course, cancer, where pre-malignant or malignant change leads to proliferation going on all over the tumor, as distinct from the very precise patterns of proliferation in the normal tissue. So the basis of regulated cell proliferation are stem cells. And for those of you who have perfect memory and total recall, and remember that I gave a lecture here in 2003, I'm going to borrow some of the illustrations from that for a few slides, such as this one. So if you have perfect memory and total recall, please look away now. If, on the other hand, like me, you wouldn't be completely sure if you were in the lecture or not, then you may wish to pay close attention to my final section on neurodegenerative diseases. But the first time I showed this slide, thankfully, before I showed it to a class of 350 first-year undergraduates, I spotted it had a typographical error. The typographical error was unfortunate. I'd missed out one letter. And instead of telling you that immortality is a rare privilege, I was about to tell 350 first years that immorality is a rare privilege. It's equivalent to an equally unfortunate error that I hesitate to describe with the Vice-Chancellor in the audience, as his previous role was as head of the Medical Research Council. And the Medical Research Council unit that my wife did her doctorate in was called the Medical Research Council Unit for Metabolic Research. And when their headed note paper was first delivered, it took a month before anyone spotted that the headed notepaper really said Medical Research Council unfit for metabolic research. But stem cells are an important component of understanding how patterns of cell proliferation are regulated. And a key feature is that only stem cells are immortal. Only the stem cells last for the duration of our body, with the exception, of course, of nerve cells that I've told you are also immortal, fortunately, for most of our lives. But stem cells have the ability to self-renew and remain in the tissue throughout our life. They divide quite rarely, which retains their genetic integrity. They don't have to suffer the mistakes of copying their DNA too often. So they divide very rarely and produce committed progenitors, which do most of the work of tissue renewal. These committed progenitors will only last a limited time. It can be a very long time, but they last a limited time, and they can divide to self-renew and to produce many, many differentiated, specialized cell progeny. But only the stem cells are fully immortal. An illustration of that, so the question then arises of how do you get variations? Well, in the case of red blood cells, which you'll notice do not have nuclei, they've thrown them away, but red blood cells can be increased in number by stimuli such as high altitude. And so the reason why athletes train at high altitude is it stimulates red blood cell production via a hormone or growth factor called erythropoietin, which, of course, in the year of the Olympics takes on a more sinister application because direct injection of erythropoietin improves stamina of athletes by allowing them to continue aerobic activity for longer. So high altitude can induce erythropoietin, which in turn induces the production of red blood cells. I'm going to show you another illustration of how a growth factor can affect growth. It's quite a dramatic one. They can have dramatic consequences, as you can see in these cabbages. These are the normal cabbages. These have been treated with a growth factor called gibberellic acid. They're rather different. So growth factors can have profound consequences, can have profound effects on form and cell number and shape. But patterns of cell proliferation, I've told you, are tightly regulated. And this is a marker that will feature again in a moment, which we've used to stain a section of normal uterine cervix, where the surface is up here, and the stained cells are ones that are proliferating down here in contact with this lower layer. And it's contact with that layer that allows them to proliferate. So there are precise geometric patterns of where cells can and cannot divide. And these, of course, provide niches in which cell proliferation can occur, and niches in which stem cells can persist to supply further cells thereafter. This, of course, breaks down in a premalignant or malignant condition like this premalignant change of the cervix or this malignant adenocarcinoma of the colon. So patterns of proliferation care about what the neighbors think. They occur at places where growth factors can be handed to them directly to regulate their proliferation. And this is a gross oversimplification, which I'm not going to take you through, which I hope will lead to relief throughout the audience. It's a gross oversimplification of ways in which growth factors can regulate cell proliferation. Any of you who feel deprived that I haven't talked you through this, I'm lecturing to first-year undergraduates on this very subject starting next week. But it's complex, and for obvious reasons I'm not taking you through it, but complex signaling pathways balance proliferation of cells but through growth factors and receptors on the surfaces of those cells, and then whole cascades of information transduction mechanisms that carry the signal on into the cell interior. I'm now going to move to what goes wrong when the balance of cell division and cell death is perturbed. And I've mentioned that cancer arises from too much cell proliferation and not enough cell death. And I've illustrated that cancer cells break the rule and proliferate much more than their normal counterparts. So what is controlling them? Well, normally, cell renewal would be regulated by two classes of genes called oncogenes and tumor suppressor genes. Oncogenes control cell proliferation by switching it on. They're often overactive in cancer cells, and overactivity can have unfortunate consequences like this train accident in a Paris station. So oncogenes control cell proliferation by switching it on. What about tumor suppressor genes? Well, they act as the handbrake on the system. Tumor suppressor genes control cell proliferation by switching it off, and they're often inactivated in cancer cells. Examples include BRCA1 and BRCA2, such as is shown on the cycle path seen here. These are tumor suppressor. This BRCA2 is a tumor suppressor gene that maintains the genome integrity and prevents cells from continuing to proliferate while they're damaged. That's an oversimplification, as some of you will recognize, but it's a deliberate oversimplification. So, if cancer cells break the rules by continuing to divide when they shouldn't, a process referred to as transformation, that's not enough, because I've told you that cells die in large numbers, and the cancer cell that dies is good news. It's not going to cause a problem, provided that it dies relatively quickly after it's appeared. The problem is that cancer cells become immortalized and can evade death. They can do this either by arising from a stem cell or by reactivating stem cell pathways so that they remain immortal and don't die out when they should. It's a major component of the problem of cancer. Ironically, cells can either be killed by viruses or, perversely, immortalized by viruses. That cells are killed by viruses is something you all know. So one poliovirus can produce 100,000 new virus particles from only one infected cell in only 8 hours. Influenza killed more people in the winter of 1918-1919 than all the bombs and bullets of World War I put together. Yet, perversely, some viruses can immortalize cells. And the notorious ones of this are illustrated both in the photographs there, plus in this very good read describing the immortal life of Henrietta Lacks. Henrietta Lacks is known in most laboratories by the name of the cell line that was derived from a tumor of her cervix, the HeLa cell line, which is estimated to have been grown in laboratories in many millions of metric tons. But the striking thing is that her cells have been grown in laboratories for 60 years beyond the date of her death. So the essentially immortal cancer cell has continued to proliferate long after Henrietta Lacks herself sadly died. So a question which may have occurred to some of you by now is, if cancer cells are immortal, why are they more vulnerable to anti-cancer drugs than their mortal neighboring normal cells? Surely they should be more resistant to them, not more sensitive. The answer to this is how they would react in a minefield. Normal cells have checkpoints that stop damaged cells from making DNA and dividing until that damage is repaired. But cancer cells have arisen by switching off the regulatory mechanisms that protect the normal cell from making that mistake. So they charge headlong into the minefield where normal cells would fear to tread. They keep proliferating and accumulate so much damage that it kills them. So that is the basis of much chemotherapy. But I want to quickly look at two aspects of translation and to introduce the perils of translation. Some of you will have seen this road sign before. It's from the border of an industrial estate and a residential area in Swansea, and the people making the road sign did not speak Welsh, so they sent off an email to the translation office to get their message: no entry for heavy goods vehicles, residential site only. They got back an email, printed it on the road sign, put up the road sign, and were then rather surprised when someone pointed out that it says I am not in the office at the moment, please send any work to be translated. There are perils of trying to translate, even when translating science for patient benefit. So I told you about these enzymes with frightening names, the DNA helicases that unwind one strand from another, and the DNA topoisomerases, which remove the helical stress that arises from unwinding them. I did this because these enzymes turn out to be extremely important targets for cancer therapy. So the drugs irinotecan and topotecan attack the type 1 enzymes that cleave one strand and pass the other strand through it. Doxorubicin, epirubicin, etoposide, and dornarubicin attack the enzymes that cut both strands of the DNA and pass another double helical molecule through it. And in so doing, they prevent the cell from synthesizing DNA, they jam the works, and the cell is unable to go on proliferating. Whereas normal cells would pause, repair the damage before going on. So the second example of, I'm going to go back, the second example of exploiting knowledge of these enzymes concerns the DNA helicases, which unwind one strand from another. And we've exploited reagents made against these to see if we can improve a public health measure, namely the cervical smear test, which is an extremely important public health measure. It saves, its current estimates are that each year's smears save the lives of 4,000 women in Britain alone, and of course very many more worldwide. But if we could improve the reliability, we might be able to save, it might be able to be even more effective. This, my pathologist colleague, Nick Coleman, who's sitting near the front, assures me this is an easy smear to read. He assures me it's very easy to spot the abnormal cells in this region. But the problem I find is they're the same color as the normals, and they're just subtle differences in shape. And this is an easy one. Heaven help us trying to read a difficult one. But if these frogs have subtle differences in shape, but the one that's a different color stands out immediately. So if we could stain the abnormal cells a different color, it might make it easier to detect them. The way we can do that is using an antibody raised against the proteins that unwind one DNA strand from the other. They're called MCM proteins. They have more frightening names than that, but I shall call them MCMs. They were the markers that were used to detect the proliferating cells here in colon. And this is the same specimen I just showed you, but the abnormal cells are stained a different color using an anti-MCM antibody. If it's done by fluorescence, it's even easier. The normal cells have red nuclei, the abnormal cells have yellow nuclei. So it can be relatively simple to detect them. And the principle here is that in normal cervix, only the deep layers should be proliferating, and the smear test only samples the superficial layers. But as pre-malignant change, and then malignancy advances, so the cells, more and more abnormal cells making DNA, are found up at the top. Our proliferating cells are found at the top. So that's the rationale. It does work. That's what I showed here. this is a section through normal cervix where this is the surface up here sampled in the smear test. This is where proliferation, where the marker is concerned, and this is a high-grade premalignant lesion with cells stained right up at the surface of exactly the sort that the smear test is designed to look for. Similarly, Nick Coleman and his colleagues have extended the study to try to improve the detection of colorectal cancer by detecting premalignant cells on the surface of stool specimens. So this test then is looking for cells that are making DNA in the wrong place. And it has resulted in one commercial product to date, produced by Becton Dickinson in the USA. But clinical trials are in progress to try to develop another reagent, which is aimed specifically as a frontline smear detection test, and we should hear from the FDA, hopefully this year, whether or not that has obtained FDA approval, which would greatly simplify its introduction globally. So my final point is to look briefly at what happens when there is too much cell death and not enough replacement by new cells, neurodegeneration. And I'm going to start by introducing you to neurons with the aid of a remarkable video which happens to have been made by my old supervision partner from my undergraduate days. His name's Brad Amos. He co-invented the confocal microscope, and he's now invented an extraordinary new giant lens. And this is looking at nerve cells stained green, accessory glial cells stained red, in a whole dish of cells. The blue are nuclei of cells. And this is a whole dish of cells looked at subcellular resolution by a single image from a single lens. It's a quite remarkable new advance in microscopy. But what I'm trying to draw your attention to is the beauty of neurons, which, as these, of course, are the cells whose death contributes to neurodegenerative diseases, such as Alzheimer's disease, of which there are 450,000 cases, patients in the UK, Parkinson's disease, where estimates vary. I've put in the minimum number of 127,000, but it looks to me as though estimates of nearer 200,000 may be more accurate. Huntington's disease is about 6,700. CJD is about 100 new cases per annum, and variant CJD associated with bovine spongiform encephalopathy is approximately 1 new case per annum. But what do these diseases have in common? Well, all of them are caused by misfolding of proteins, resulting in the death of nerve cells. So proteins should fold into orderly structures. None of these proteins is one that goes wrong in a neurodegenerative disease. This is pure self-indulgence. These are proteins my lab has worked on, and I still think of them as things of beauty. They should have very precise folded structures, as illustrated here. But as proteins are degraded, fragments are generated, and some fragments form insoluble aggregates that can kill cells, kill nerve cells. These can be intracellular, as seen here in this artist's impression of nerve cells showing brown blobs as intracellular protein aggregates, and these are common to many of the dementias. Or they can be extracellular, like this amyloid plaque from Alzheimer's, which has both intracellular and extracellular complexes, though of different proteins outside the cell and inside the cell. So it's the formation of these insoluble aggregates arising from misfolded proteins during protein degradation that kill the neurological cells. One thing that can bring possibility of hope for these diseases are, of course, neural stem cells, which raise the possibility of being able in future, at some point in the future, to be able to replace some of the cells which have been damaged by either intracellular or extracellular protein aggregates causing those neurological diseases. This is becoming increasingly plausible as an approach. for the immediate future, though it is not in significant clinical use at the moment, but trials are beginning to get off the ground. So in this lecture, I've tried to infect you with enthusiasm for the beauty and the fascination of living cells. I've given you a glimpse at how cells divide, stressing the importance of condensing DNA into chromosomes and then pulling them apart on microtubules. I've argued that cell death can be good for you, and that it is a very dramatic process that results in total disintegration of a cell to ensure that wounded and damaged cells do not persist to threaten us with cancer. I've argued that cell renewal is extremely tightly balanced to generate a precise match between cell production by division and cell death. And I've argued that cancer and neurodegeneration sadly arise through faults in the process of balancing cell renewal and cell death. All of my research has been was supported for 45 years by either of these two funding bodies, Cancer Research UK or the Medical Research Council. But I want to finish on something slightly different, with one more slide for a slightly different point, which is it's exactly 30 years since I was approached by a former fellow of Darwin to ask if I would be interested in fellowship in Darwin. And that individual did more to exploit knowledge of cell immortality for patient benefit than anyone before or since. It was Cesar Milstein who invented monoclonal antibodies. And Cesar did that by fusing an antibody-producing cell, a lymphocyte, with immortalized cells that persisted in culture indefinitely. Cesar found that by fusing the two, he could obtain a hybrid cell which would continue to produce pure antibodies essentially indefinitely in culture. That is now the basis of a massive multinational industry and has had enormous impact on medical research, and in particular is now having impact on medical practice as therapeutic antibodies as well as diagnostic antibodies become increasingly used in medicine. So I would finally like to dedicate this lecture to the memory of Cesar Milstein. Thank you.
Epidemic apoptosis. We retire. We're retiring simply because we've got to the age of 67. One reason to be grateful that this is happening is that from now on, roughly now on, academics will have to decide when to apoptosis each other in order that the University of Cambridge can continue to be healthy and active. And I wondered by what process cells do this. Then he showed his wonderful diagram, so complex he didn't even start to describe it, but I understand we can go to lectures in the next 2 weeks. And I realise it's a model of the university committee structure we're about to well, I just found that wonderful. We are made of information, but how delicate and how intricate, how ingenious and yet how robust. It was a fascinating talk, Ron, and a very fitting tribute to Cesar Milstein. Thank you very much.